So the use of carbs and lipids in energy storage, the advantages of lipids are lipids contain more energy than carbs, so storing them is lighter. Lipids are insoluble in H2O, so there's no problems with osmosis in cells. The advantages of carbs are that they are easily digested, or easily digested than lipids, so energy stored can be released quickly. And carbs are soluble in water, so they're easy to transport and store.
There's two different types of proteins. So there are four main functions of proteins: structural, transport, movement, and defense. An example of structural is collagen, which is a fibrous protein. It strengthens bones, tendons, and skin. An example of transport would be hemoglobin, which is a globular protein. It binds oxygen in lungs. It binds oxygen in lungs and transports oxygen to organs, to your vital organs. An example of movement would be myosin, which is a fibrous protein. It causes contraction of muscle fibers, which then causes movement. And then an example of a defense protein would be immunoglobin, which is a globular protein. Its function is to act as antibodies to protect your body against disease.
Okay, so we're outlining the role of condensation and hydrolysis in the relationships between dipeptides or amino acids, and that is polypeptides plus water equals that, and that would be hydrolysis, and condensation would be dipeptides or amino acids minus water equals polypeptides. Same would be with polysaccharides and water equals disaccharides, which is hydrolysis, and the reverse of that is hydrolysis.
So on the board right now we have three functions of lipids. As we all know, lipids are fat. So the first function is energy storage. Lipids store energy in the form of fat in humans and in the form of oil in plants. The second function is heat insulation. A layer of fat under the skin reduces heat loss so we can stay warmer. And then the third function is buoyancy. Lipids, since they're less dense than water, it's easier for animals with more fat to float in water.
So, polar and nonpolar amino acids, controlling the position of proteins in the membranes. For polar amino acids, parts of proteins protrude from the membrane, and nonpolar amino acids, they cause proteins to remain embedded. Creating hydrophilic channels through membranes, polar amino acids, the amino acids create channels, and in nonpolar, there are hydrophobic R groups.
This is amino acid. This is ribose. This is glucose, and this is a fatty acid. Okay, the three examples of monosaccharides are glucose, galactose, and fructose. These are the disaccharides, which are maltose, lactose, and sucrose. And then the polysaccharides all the way on the right are starch, glycogen, and cellulose.
Organic and inorganic compounds, and carbon in living organisms makes up an organic compound, and there are three types of compounds, which are carbs, lipids, and proteins. And inorganic compounds have no carbon, and some of the...
So on the board right now we have the function of glucose, glycogen, and lactose. So on the board right now we have the function of fructose, sucrose, and cellulose in plants. The function structure is the number and the sequence of amino acids in a polypeptide. Basically a regular repeating structures, including beta pleated sheets and alpha helixes, stabilized by hydrogen bonds between groups in the main chain of polypeptides.
The structure of our protein is a 3D conformation of the polypeptide, and this form of the polypeptide folds up after being produced by translation. The conformation is stabilized by bonds, which are ionic, hydrogen, hydrophobic, and disulfide bridges. So the quaternary structure is the linking together of two or more polypeptides to form a single protein, and for example, collagen is 3 polypeptides together, and that is 2 polypeptides becoming 1.